Good afternoon, everybody. It has been a long day, and I thank all of you that have decided to stay to hear this one last talk. Wendy and I spoke at the World Orphan Drug Congress earlier this spring, and one of the concepts that emerged from that talk was that of a table. So as we talk this afternoon, you will see where we got this idea from and how it all comes together. So, Wendy and I are the co-chairs. And the members of our panel are Steve Groft, Hudson Freeze, Matt Wilsey, Jane Gershkowitz, Jacob Jacoby, and Kate Rowan. And they will each tell you a little bit of who they are and what they do when they're introduced, and I won't duplicate all of that now. So the first thing we want to talk about is the changing role in pharma. I've been in pharma since 1999, and over the course of the past 15 years, I have to say it has changed a lot. Jane and Steve Groft are sitting here with me today, and we all met at a NORD meeting in 1999 in the fall in DC, and there was 15 of us in the room. Also, Nancy was there. That was the whole community for orphan drug development. Look at how many people are here today. And look at how many meetings you get invited to during the course of the year that are orphan drug focused. So you can see it's changed. So pharma, traditional pharma, has moved from working on drugs for large patient populations to working on drugs for small patient populations. When BioMarin developed Kuvan for PKU, there's about 1 in 10,000 to 1 in 20,000 Americans have PKU. That's a lot of people. The company I'm working at now is developing a drug for maybe 100 patients in the US. So the sizes have changed. The high cost of drug development has done nothing but go up because more and more is being required in order to prove that your drug works. And in the case of very small patient clinical trials, multiple sites are needed because each site might only turn up 1 or 2 patients. So you might end up with 40 or 50 sites to get 100 patients in a study. Regulatory approval is now required in multiple countries. I mean, it used to just be you got your approval in the US and didn't worry too much about the rest of the world. Now we're busy talking about getting drugs into South America, into Asia, into China, into India, and all of those countries have their own regulatory approval processes, which add considerable time, effort, and money to the situation. And then you have the sales force distribution challenge. So if there's 100 patients in the United States, where do I put my sales force? How do I get drug to them? If there's 1 or 2 patients in a country in Europe, which isn't unlikely, what do I do for a sales force? All right, come on. It's not advancing. Okay, during the course of this past 15 years, things have changed in the patient community too. Today, I would venture to say that almost every rare disease patient has a PhD from Google University, right? Where do we spend our lives? On Google. What do we know? Anything Google told us. Also, with the rise of social media, patients are much better connected. They're in closed Facebook groups, they Twitter, they call. The cost of long distance has dropped dramatically in 15 years. And they have also become fundraisers, and they are raising money for their own research projects. And that's also a big change because now with them raising money for research projects, they want a say in what's being done by the company. Oops, sorry. And things are changing in academia too. Universities have become very interested in licensing their technologies. They see it as a huge source of revenue for their investigators and for their other programs. The researchers are also busy looking for funding, and as we heard earlier, funding for NIH does not always increase year by year, so that makes the competition a little more stiff. Academia is also a source of key opinion leaders who are the physicians you want to lead your research study because they are the ones that know the most about the disease. And also most universities have clinics where patients with rare diseases are seen. So I've identified the key players. They all need a seat at our table. It's important that we all understand the roles that we play in this process, and they are all different. It's vital that we communicate with each other, and if we do, success is possible. So, that's not me. Our format for today is that everyone will give a brief overview of what their role is and why they have a seat at the table. We're going to do a case study that's going to talk about the role everybody would play if a patient comes to you that has a rare disease and wants access to a drug that's in development. What are the challenges in those roles? And for all of us, the challenges are very different. There is a new movement afoot nationally. It's on the ballot in Arizona, where I live this fall. It's already been approved in Colorado, and it's called the Right to Try law, which is a law that was passed in Colorado that says that patients have the right to try any investigational drug, and that the physician in the hospital or clinic that administers the drug is not to be held liable for any problems that occur. That sounds great. But what it doesn't have in the fine print is that pharma is not required to supply the drug. So you can have the law in the book, but a pharma company does not have to give you the drug to try. And finally, we're going to have a couple of examples of successful collaborations between patient groups and industry. So, Jane, do you want to kick us off?
Yes, so we have a table in the front that we have an even-sided table, and that this is sort of representative of the fact that everybody sort of has to have the right seat at the table, that all the legs have to be even, and that one side would represent academia, one side would be industry, one side would be government, and one side would be patient advocates. This is really different, right? Because I don't think that patient advocates had a seat at the table just a little while ago, but with that seat at the table, what does that mean for everyone else? So what I'd like to do is just to maybe go down the row here, and thank you, Nicole. Now that everybody has that mental image, we can just move on. So here's what I'd like to do is maybe everybody can just introduce themselves so we know who's talking.
I'm Jonathan Jacoby. I am with the Hide and Seek Foundation and a collaborative project of researchers and parents of children with Niemann-Pick type C.
Thank you.
Am I mic'd? Oh, perfect. My name is Kate Rowan, and I'm a professor at the University of California at Davis in the Department of Pediatrics, Chief of Genomic Medicine. And my role is an academician. I'm a physician scientist, so I actually kind of have the best of both worlds in academia, where I see patients, and I have a research lab as well, and study the group of syndromes called RASopathies.
Hi, everybody. I'm Jane Gershkowitz. I'm Vice President of Patient and Professional Advocacy and Public Policy at Amicus Therapeutics. I've been with Amicus for over 8 years. Came to the industry side after being Executive Director at National Tay-Sachs and Allied Diseases. I was there for 8 years as well. And at that time, really worked to bring the organization to its next level, where then I was happy to turn it over to Sue Khan, who's here with us today. And I think one of the main things that we did during my tenure there was not only to work on board development and fundraising, but particularly research. And as one of the older rare and genetic disease patient organizations in the country, National Tay-Sachs always had done research, but we overhauled that system to bring in money for seed research, for our own grants, but also we developed a collaboration with the National MPS Society that was joined by some private family foundations in the MPS space, and we brought that to NIH. And with NIH's funding, we actually had the largest amount of funds put together up until that period of time to study lysosomal storage disorders affecting the CNS. My background also includes marketing communications in the nonprofit as well as for-profit side, so I have a fair amount of experience in community health programming, health services, mental health, home care, adoption and immigration services in doing marketing and communications for that. And all of that has really come full circle. Now, as I work for an emerging biotech that has just received positive data from its phase 3 studies for its lead asset, and now we are moving forward with submission. But our work with the patient community throughout all of this, not only for that product, but in general, has been to understand what the patient community goes through. And I think that it's been really, it's really been an honor and a joy to be able to have a foot in both communities, in industry and in the patient world, to be able to help a company that has its heart in the right place while seeking to be successful for its investors. And look forward to talking with you about that.
Am I alive?
You are.
Yeah, I'm alive. Okay. Well, my name's Hud Freeze, and I'm a basic science researcher at the Sanford Burnham Medical Research Institute in La Jolla. And my background is in slime molds. I mean, probably two worst words you can imagine in the English language. So I understood the glycobiology of slime molds, and that's where I started. But I moved into rare diseases. And do I have a couple minutes to elaborate on that?
Go ahead.
Okay. Glycobiology. You probably have not heard about it unless you went to see the movie Extraordinary Measures, where Harrison Ford is, "What's wrong, Sally? You're not up in your glycobiology." And that's the first exposure most people have to my field. And very, very few physicians had ever had any exposure. And it turned out that there was an indicator, a biomarker, for abnormal addition of sugar chains onto proteins. But nobody knew what the basis of the disease was. And so I began working in this area because it overlapped with some of my slime mold experiments. I had mutant slime molds. So is there any possibility that there would be some connection in all this? You know what? We did some experiments in human cells, unknown disorder, but they matched the slime molds. That was really amazing to me. Then what happened is, because the sugar was deficient, it was called mannose. Maybe you've heard of D-mannose. And so that sugar was deficient, so I did the most simple-minded experiment you could. I took mannose and I threw it in the culture medium. And now everything became normal. I said, wow. Wouldn't it be something if you could actually do that with patients? Because mannose is a natural sugar. And I said, well, geez, let's take a look at the literature. There was no literature. There's no fundamental science on mannose as a nutrient, as something you could eat. So we lined up people out of my lab after getting permission from the FDA, and we sat around over a couple of weekends drinking mannose. One person got diarrhea, so we knew where the limit was. And so it was an interesting situation. 2 days after we finished that experiment, I got a call from a doctor in Germany. He says, we've got a kid. He's lost 40 units of blood. He's probably going to die. We saw your paper that talked about mannose, but do you have any idea how much mannose you could give him? I say, yeah. Give him this much, this often, he'll achieve this level in the serum. It'll be cleared out at this rate. Let me know what happens. I said, I really doubt that it's going to do anything. And you know what? The kid completely recovered. He lost almost all of his symptoms. He was more healthy than he had ever been in his life before. And you know what? We had no idea why that worked. Where's the fundamental science that's going to tell you how this happened? One of the experiments that we did looked at how mannose was metabolized in slime molds and in people cells. And we found out that this mannose was being abnormally metabolized, and what that meant was that it wasn't getting degraded. It was going into the pathway that was deficient. And so once we understood that, then we could do the enzyme assay, then we could do the gene sequencing, and we found out that was the reason for it. So it all made sense. The whole basic science, the clinical aspects, they all came together. And that was a real thrill. But it started us off thinking that maybe there were other opportunities we had here. And because, and this was really important, because very few people knew about glycobiology or how those things worked, and patients were being found that had deficiencies in glycosylation, there was no place to go. So physicians and families were forced to come to basic scientists. We had a few answers, not really very many, but that was how my involvement with families started out. And I've come to realize that our labs are only a click away from all of you. So if you find something that is wrong with your child and you don't know what the disease is, some of the symptoms match or some of the tests say glycosylation, all you got to do is do an email and you push send. And that has been fulfilling as a basic scientist. It's been revolutionary in my career. It's enabled me to connect with patients, with physicians, and discover 15 more diseases that were never known before. That's the power of the collaboration between scientists and physicians and all you folks. One thing I want to make clear, and it came up in the last presentation, is that the funding for fundamental research has decreased 25% in the last 10 years in real spendable dollars. And while we each have our specific disorders that we're interested in, we want to see work progressing, I can absolutely tell you that the fundamental science from over there may affect something sitting right next to you, your child, and you don't know where that's going to come from. And as long as we keep robust funding in the NIH, we have a chance. What's happening right now is bordering on a catastrophe. You have the power as voters, as taxpayers, to reach out to your congresspeople, to tell them the stories, and to say support not only our kinds of diseases, but the General Foundation for Research. And I'll finish off just by saying I have an example of that from my longtime past. When I was an undergraduate student, I had the opportunity to go to Yellowstone National Park to work on hot springs bacteria. Come on. I mean, who cares, right? So these bacteria live in boiling water. Well, whoop-de-doo. So I isolated a bacterium from there. That was really great. Procter & Gamble came to the lab to see if maybe they could put it in Tide to make a better, washing machine or whatever it be. And that didn't go anywhere, so it sat on the shelf for 20 years until Carrie Mullis came along and said, oh, wait a minute, I can take that little piece of fundamental useless research and convert it into something that's really important. And he did. That's the polymerase chain reaction. That was worth a Nobel Prize and completely revolutionized genetics. That's the power of basic science. And we got to keep it going. And if we don't, we're in trouble. Thank you.
Well, there we go, a fabulous story of collaboration, and started and led by a scientist, right? A basic scientist. And I think that's the power, right? The leader can come from anywhere. And it's just having the power, the foresight, the vision to reach out and reach across. So, so great. We're just going to lead off with that. But now we're going to move over. Barbara, you've talked, and
I already talked.
Steve,
Okay.
you want to follow that? Anything
It's
about
hard
NIH?
following that, I mean, HUD's a great guy. I've known him for years, and it's just so great to see the success that's coming. So congratulations, and keep up this fabulous work. And even with more NIH grants, it's even getting better. So we hope for you and others. First, thank you. This is Friday afternoon. It's 3:45 almost, and I cannot tell you how many meetings at 3:45 on Friday afternoon. It's like a disappearing factory. The veil comes over and everybody disappears. So thank you for staying, and it really does, I think, reflect the commitment of so many people to rare diseases, to your families, family members, and yourselves who have these diseases. I've had the most unbelievable opportunity you ever could ask for, and that's to work in rare diseases for about 30 plus years. starting back in 1982 when we established the Office of Orphan Products Development at FDA, then in 1989, moving down to NIH after a special commission, setting up the Office of Rare Diseases, and then we added research later at NIH, and staying there until I retired in February of this year. And I failed retirement twice, and my wife's convinced, and many others here are convinced that this will be the third time I'm failing. One of the few times in your life that's good to get an F in something you're doing, you know, and so. But it's been such an unbelievable pleasure to work with so many people. And what we've tried to do for so many years is to help establish a research agenda, help to pull together those legs that you see on that table, and to give it a surface that we all can work on. Very, very important. And I think for so many diseases, we didn't take advantage of this opportunity. I think people are realizing now that if you want to be successful with the rare disorders, you need all of the legs, you need all the people working together. And so we've utilized a number of mechanisms, including the scientific conferences, in which we involve many of the institutes and centers, the 17 institutes and centers at NIH that are involved in actual research, or 27 institutes and centers all told. So we've utilized the resources from the centers to help you and the community to develop a research agenda and identify the scientific opportunities. So many times you have to start from that little idea, that little thought, and expand it, but you have to bring in all the players. So that has been wonderful. We've established several years ago the Rare Disease Clinical Research Network, and the real wonderful thing about the RDCRN was the collaboration with multiple researchers, multiple sites, multiple diseases, but probably the most important aspect was bringing the patient advocacy groups and requiring the patient advocacy group to be part of the research network. And so I think that has opened the door to an understanding that patients and their families are truly research partners in all that we're doing in the rare diseases field. So just some of the other activities involve the development of the Undiagnosed Diseases program at the Clinical Center. We now have expanded that into 6 centers throughout the United States. We'll be traveling to Rome in September to talk about how we're going to expand this into the European continent. And then we've also already received a request from Australia to work in Asia and Australia next year, so that we hope at some point there will truly be a globalization of what we're moving towards in rare diseases, and that we can communicate, and whether it be through telemedicine, telehealth, whatever we like to call it, and work through many of the problems that we'll be confronting, having physicians see patients in different locales and what they are, several of these problems that do exist. And I will say too, I've had the unusual opportunity, Barbara's been involved in developing 2 international groups related to rare diseases. One is called ICOR, the International Conference on Rare Diseases and Orphan Drugs, and the other is IRDIRC, the International Rare Disease Research Consortia. And again, bringing the partners together, all of the legs of the table together, to focus on how are we going to really understand what's going on with rare diseases and communicate it back out to the public, to the healthcare providers, and to the research community and the industry, along with the government agencies. So I'll end it with that, but the only thing I'd ever wish is that all of you would have the opportunity to do the great things that I've been able to do and be involved in, and the other folks working with wonderful people that are up on the stage with me. It's been a fabulous experience. I think you're realizing it as you work with so many of the researchers and your families, what outstanding people you're dealing with. So I'll end it with that.
Hi, everybody. My name's Matt Wilsey. I'm going to keep it relatively short because I know I'm certainly anxious to jump right in. I'm sure you guys are too. I represent the patient advocate leg of the stool. After a 3-year diagnostic odyssey with whole genome performed at 2 top centers, and some help from social media and our friends Christina and Matt Might, we diagnosed our daughter Grace, who's about to turn 5, with a rare disease called NGLY1 deficiency. There are 13 living patients, at least, that have been diagnosed in the world thus far. We're working closely with HUD and probably about 8 other centers worldwide now. There's about 40 people working on our disease. It's now my full-time job. I started my career in government public service, first at the White House and then at the Department of Defense. And I joke that I'm a recovering tech entrepreneur. So we're really trying to apply a Silicon Valley business model to what we're doing with our foundation, the Grace Wilsey Foundation.
All right. Well, thank you guys for that, for the introduction. So I know that maybe we'll save your, Jonathan, for your case study for a little bit later. That's okay. And what I'd like to do is then think about like sort of where each person comes from. I think all of us in this room, when you think about being a patient advocate, you think that that maybe you're making some assumptions, which is that what could be a higher mission than finding cures for your children or finding cures for yourself or your family members. But I think the way that we can have true collaboration and to be effective is to really understand everyone else's point of view. Of course, that has to be the highest goal, but the people who have jobs and their own families also have individual career goals and have to think about where they're coming from. And I think the more that we understand what makes everyone successful, the more that we're in the right place to create win-win scenarios and to be able to reach across the table and figure out how to make the most effective thing. So let me ask each of you first, what from your role as, for example, an academic, what do you bring to the table?
So, with regards to academics, just like what Dr. Fries was saying, is that what we focus on, especially from a clinical translational point of view, is we see patients in the clinic. As an example, and kind of to tell you the little bit of odyssey that led to why I'm sitting here today, I guess, is with regards to my interest as a fellow in medical genetics in Costello and CFC syndrome. These are two very rare syndromes that turned out not to be so rare after all, because as we were identifying over the course of the years what the genetic etiology was for these particular syndromes that were at one time considered rare, and I'm not going to call them rare anymore, is that once we found out what those genes were, we actually learned from an academic point of view. You know, we start off in the clinic, we see these patients, and then we try to identify what are these genes. And this was, trust me, well before next-gen sequencing. So now a medical geneticist, and a medical geneticist is an individual that pretty much we're the last ones that get called into the party in trying to identify what is going on with this patient to try to find a unifying diagnosis. Usually the cardiologist will see him first, and then the pulmonologist, and then the renal doc, and infectious disease, and all these people. And then when they can't figure out what's going on with this patient, they get sent to the medical geneticist, and that's me. Well, in the olden days, we didn't have next-gen sequencing. We weren't able to sequence the exomes and try to identify what the genes were at that time, kind of in a shotgun approach. We actually had to think about either by positional cloning or by trying to do your best guess what was going on. So as an academician, it was my job to try to figure out what is causing, what are the genetic reasons, what are the genetic mutations that are causing these particular disorders. I actually worked very, very closely with advocate groups, and I can tell you that when we were doing this almost 10 years ago, in my mind, it was not an unusual thing to do, as these advocate groups, as I tell them, you know, because they're parents of individuals with special needs and more than likely a genetic disorder, even though it may or may not be diagnosed, they truly are honorary medical doctors or honorary MDs. They know more about their children than any of the doctors that they will ever go to. And so as an academician, what we bring to the table is, number one, trying to identify what's going on, trying to provide the best practices, the best medical care that we possibly can for these patients. And to work, I feel it's really important for the academicians to work very, very closely, not only with the parents, but with the entire advocacy group as well. So, so as an academician, my job is actually to provide the best practices, to try to identify what's going on. And once we identify what's going on, hopefully bring together some kind of treatment, whether it be moving towards clinical trials or trying to find the best mode of treatment for that patient and patient group.
And let me just have a follow-on question, and that is, how do you balance the need to publish, right? Isn't that one of the criteria for being a successful academician with the demands and I'm sure the desire to make a difference to your patients?
I think I was really lucky in that it kind of went hand in hand. I do have to say that when we published our finding for cardiofacial cutaneous syndrome and identified the genes, it turned out to be genes for that. The reason why it actually ended up in the journal Science, which is probably one of the most prestigious journals you can publish in, was because we identified that it was an entire pathway. It was a super important, critical cancer pathway called the RAS-MAPK pathway. And it's not very often when you publish the identification of genes, it's not very often in the same publication that you can write, you know what, there are actually treatment out there. This is a really well-known cancer pathway. There's already small molecules that have been developed that companies have in their backhand, in their pocket, that they could apply as a treatment in the small molecules to RAS and to the MAPK pathway. And so as an academician, yes, we do have to publish. We have to write grants, which I have a grant due in just a couple of weeks. But I'm sitting here because I think this is really important to talk with families, to get the story out, and to move towards clinical trial and to move towards treatment. And I myself have been to and spoken to many, many companies, of which probably many of you own stock in, and have been told, you know, this is probably the most fascinating story we have ever heard. Yes, you're absolutely right. We have small molecule inhibitors that target specifically this pathway, but we're not going to release them to you because we don't want to see it fail. We don't want to see it fail before it gets approved. And so that's been the big roadblocks that we've had in academia. I know that's a little off topic from publishing and writing grants, but I think I've been very lucky, and it's been hand in hand, and it's been a very productive relationship with advocacy groups in academia.
Yeah, actually, I think that's a perfect transition, Jane. Is thinking about, that was actually going to be my next question of what are the barriers for thinking about how to bring drugs to market. But maybe before we go there, Jane, if you want to talk about what does industry bring to the table and what makes you successful?
Absolutely, thanks. So as far as, I just told you earlier a little bit about my background. And so in industry, particularly in orphan product development, I think what we bring to the table, not all companies, but more and more companies every year are truly seeing the value of the collaboration with the patient community and patients as partners. At Amicus, I'm very fortunate that that's been one of the main focus points of the company from the very beginning, because our CEO has 2 children with Pompe disease, and so this has been his passion. And Dr. Fries referred to the film that was made on John's family's life, Extraordinary Measures. And so the fact that the patient perspective is considered as important as the science, as the commercial development, as the clinical trial program is key. And we're able then to value that relationship from the early days compared with some companies, particularly larger pharma companies, that may not look at the patient community until they're into or through phase 3. They're concerned about expectations that are being managed, as we are, but we feel that if you start a relationship early on in phase 1 and you're able to educate patients and answer questions about the technology you have and what you think it will do, and you are honest and people have a good understanding of what you're trying to do on their behalf, and that's the role of the patient advocacy person or team in the patient community, is to help educate as to the technology and the hopeful path forward, and to help manage expectations so things are realistic, and that within the company, you're the patient community's representative, so that in the development of protocols for clinical studies, you have a sense, based on what you've learned from your partners in the patient community, what are the unmet needs? What are the things that patients and their families are willing to do to participate in a clinical study? And you pour that in cross-functionally to the development of a protocol for a clinical trial. Because, you know, we know that there's this urge that just gets bigger and bigger. The more questions you ask and the more you think you might be able to get out of something, more questions follow on. But you can't necessarily do that in a clinical trial. You can't ask people to come in 5 times a month, for example. You can't, you know, when you're thinking about biopsies and you're thinking about poking and prodding, there is lots of questions that you might want to ask, but what's realistic to ask somebody to go through, particularly if you're needing to enroll a study? And we're talking about studies that are hard to enroll anyway, because we're speaking about very small patient population numbers. People are spread out literally all over the globe. You're not going to find a concentration in any one region or any one state, let alone any one country. So we really represent the patient community within the company and the company to the patient community. And this partnership has served us very well, and we see the model from the rare disease community now starting to expand outward to other members of our industry. So the words of, you know, patient centricity and all of these catchphrases that are really hot right now, those of us who've been sitting here and have known each other forever, we've been living that for a very long time. The one other piece that I think it helps with very much is when we might have something on a shelf that we're not going to do anything with commercially, but we know it has potential value, and then working with the patient community to see if we can come together in some agreement so that somebody else might be able to take it forward. So I mentioned earlier the work that I did at National TASACS in terms of our research collaborative. The IP lessons learned then, having been at the earlier days of our research program there as a patient organization funding. So now that I'm on the other side and I'm working with patient organizations or family foundations that are giving us funds to either help with the very early program, or we're looking at licensing one of these, you know, shelved compounds, has helped enormously. And I'm able to work with my legal team and my science team so that they can understand where the family is coming from, which of course is from a point of passion. And as Pat said, and it was quoted this morning, you know, we're all impatient patients. They need to understand that. But I'm also there to help them understand what's the reality of timelines and time frames and the legalities, and again, how we can all come together and collaborate so that everybody, and compromise, because compromise is a part of all of our relationships, and how we can move things forward for the greater good for everyone. So I think that that's a piece that's very important, and if any of you are working with a company or thinking about approaching a company, I urge you to see if they have a patient advocacy function, because that may be, as much as the scientists are awesome guys, we want them to keep working in the lab. And we will work with them and we will work with you. Years ago, I actually had a very well-known stem cell scientist say to me, you know, every day I get somewhere between 3 and 5 calls from families wanting to know where I am on my work for their child's disease or a disease that has some potential crossover. And as much as I love talking to families, I can't get my work done in the lab. And so at that point, this gentleman said, as the patient organization, can you please be the point person and make sure that my updates and my information is getting spread to your members? And so, it's very analogous to what I'm doing. So I'm the point person, or other members of my team are, for anybody in the patient community that's thinking about, or we're already engaged with in terms of an early program. So I hope that that's helpful and answers your question.
Yeah, thank you. All right, Steve. So we heard a little bit about what you bring to the table, but maybe you could elaborate on that and what it is that makes people in the government, and maybe you can...
We don't know what makes people in government now. All kinds of things, yeah. That really, I think it does lead to success. But I think when you think of the NIH, you look at the budget, and HUD mentioned, you think $31 billion per year, $30 billion per year. It's a lot of money. How do you extend that over to these 17 institutes? And some of these institutes are funded, to a greater extent than others. And so we trying to bring an emphasis to rare diseases, and we've had to do this through a systematic approach because there were first so many disorders, so many people coming in asking for money, that we really had to put into place processes that folks could work together and figure out what are the programs that are really necessary to move the research and discovery forward. And so today, where we see a tremendous emphasis coming on translational research, where we take the discovery and hopefully move to the intervention. And I think one of the things we tried to do is introduce, especially the patient advocacy groups, and what we often forget are the scientific and medical advisory board members to have an understanding what is available. There are so many resources available at NIH that we often get lost in them. I think part of our task has been to try to open those resources up and to show people the path to obtain the resources and to really get an emphasis on their disease. And when we're successful, and you've heard of several successes here at the meeting, it's amazing and it's wonderful because you know that you're moving towards treatments. But it doesn't just happen in a year or two. HUD's research and Kate's research, for all those years, things were developing slowly, and it takes time. Same as developing a product. It takes time. It takes money. You have to tie the pieces together. I think that's the hardest part for people to really understand and sort of figure out, how do we bring all the pieces together? And that's what we've tried to do in the office. That's what we try to do at the NIH. And I would say to all of you, if you don't know who at NIH is focusing on your disease, you really should know that. And if you want to call me, I'm now a senior advisor to the director of NCATS, the National Center for Advancing Translational Sciences. So I can talk about a lot of things, as I do, as some of you know. So we have the opportunity to sort of guide you, and I can definitely refer you into the Office of Rare Diseases Research if you're looking for a project on the Therapeutics for Rare and Neglected Diseases program. So there are many programs. If you have a neurological disorder, the NeuroNext program at Neurology Institute, the NINDS, wonderful programs. But sometimes I think people don't understand what is available, and it's a huge place, and it's really easy to get lost there, and that's what we want to try to prevent you from doing, but yet to open those doors for you. So we'll end it with that.
All right, thank you. And finally, Matt.
Well, I probably don't need to tell what all the patient advocates in this room bring, but really, I think what's the primary thing is passion and determination and a lot of courage. And in our experience, I'm sure it's true for a lot of you, and I know there's some people who have children who are undiagnosed in this room, is that determination absolutely leads to a diagnosis. It might take time, but without really that passion, the researchers can pick up hundreds of different diseases. HUD doesn't have to work on NGLY1. He could work on 100 different things. He chooses to work on it partly because of the passion from the parents. And it's an interesting scientific case too. More practically speaking, I think one of the things that parents and advocates and patients themselves bring is really filling the gap, finding those pieces. So scientists are very busy, right? So I can't call HUD or call another researcher and say, hey, I've been thinking about gene therapy. Who should we contact? Can you contact them for me? Can you figure out who the right people are and then give them an email or a call? That's our job. And so a lot of what I spend my time every day is looking at the board, seeing where the gaps are, and then sort of triangulating, okay, who's the best at gene therapy? Who's the best at IPS lines? Who's the best at mice? Who's the best at zebrafish? Who's the best at worm? And then finding those people, vetting them, knocking on the door, convincing them to join the team, and then taking the time to assimilate them to the larger team in a way that's really effective. You can't just recruit somebody and then drop them in and be like, hey, Hud, I want you to work with this guy now. Great, good, see you, I'm on to the next thing. It's a hand-holding process like any courtship and relationship. And so I think that's another thing that we all bring to the table. And if you are diagnosed and you're working towards a treatment, or if you're undiagnosed, you are the clinician. You are with the patient or the patient themselves 24 hours a day. The clinician only sees the patient for 5 minutes to 60 minutes, maybe if you're lucky, but you're seeing subtle variations. And if you're on a drug or trying a different trial, you're going to pick up on the little clues, and you can relay that back either to the researcher or to the clinician themselves. And that information is tremendously valuable. So I think these are all things that we can do, practically speaking, that has a lot of value.
That's fantastic. So before we jump into maybe some more the harder questions and more controversial questions, maybe Jonathan, can we hear about your incredible work with your successful collaboration around SOAR?
Well, I think so much of what you've done already described, I can just say, apply that to Niemann-Pick type C, except for that we knew what disease my son had already. And so many of you on this panel have helped me get to, or helped us get to that place. I think what happened with my son and with us is that we found out that our son had a disease, a very rare disease, very early on. And there was a group of scientists that was working very hard and trying to understand the disease, but they were all basic scientists. And they were all inclined to try and find out more about the disease. And they had contact with the patients and with the parents, because it's mostly a pediatric disease, but there wasn't a lot of, there was very little parental push. Maybe outside of one or two families, the parents were waiting, and that's one of the things that happens, especially with a disease that's a serious disease and where the children are affected pretty early on, and they take a lot of care and they take a lot of time, and the parents, by the time they get to even thinking about helping their children, are too tired. I mean, they just can't do anything. And I was very lucky that we found out about my son when he was almost completely asymptomatic, and I felt fortunate that I could give the time and the energy to something without having to worry about, changing his diapers or helping him walk to or helping him eat or anything like that. So that was one thing is the idea of bringing that energy to the scientists. And then, and I think this is one of the key things, providing them with incentives that were at least as strong, if not stronger, than the other incentives they already had. So you talked about publishing. Publishing is an incentive. There are some in the patient advocate community who get angry at academia because why do you always have to publish? Why do you always have to publish? Well, they have to make a living. And you have to make a living, you have to publish. Otherwise you don't get promoted, you don't get a tenure, you don't get to do your work. So we have to understand that and provide other incentives. And usually an incentive can be monetized. And so what we did is we found a group of scientists who were all basically doing, or essentially doing, basic science, and we did a couple of things. One, we made sure that they knew the kids. We made sure that they were scientists who already had interaction with children who had NPC. Second, that they had a collaborative inclination They were willing to share information, and they understood that that had to be something beyond presenting an abstract at a conference. And I think there's something that it's hard for me to characterize, but there's something about sort of an attitude that was there. You could sort of, I could talk to somebody. I think I met, 25 researchers in Niemann-Pick type C within the first 2 years after my son's diagnosis. And I knew almost right away which ones were going to collaborate with others and which ones weren't. And I didn't spend much time with the ones who weren't. Or what I asked of them were things like, can I use your name as an honorary member of this project? Because that's all they were going to do. But there were others who really took the time, and you could tell, you could see it in their eyes. And I do want to say one of the things, and you've certainly done this, but I want to just put words to what you've done, which is the thing that I brought more than anything else, or I bring more than anything else to this enterprise, is what I already had, what I was already trained to do. I still am not completely sure what a cell is. I'm honest. I mean, I did not do well in biology. I did not do well in
terrorist
science.
organization.
Yes, right. That, by the way, that's what I was trained to do, understand what a terrorist cell is, but I don't understand I don't understand biology. What I do understand is how to listen to people, how to empathize with people, diplomacy, how to bring people together. And I had worked a great deal with Israelis and Palestinians and political people, and trying to get them around the table. And it was easier sometimes to get them around the table than it was to get scientists around the table, the same table.
Yeah.
But I had that skill, and that was really, really important. So the next incentive, once you had a group of people who could collaborate and who were willing to collaborate and who had a sense of connection with the families and with the children, and who were, who had the complementary skill set, right? You don't want to get three mouse people to collaborate with each other. You don't get that much from it. Once you had all that, then it was just a very practical question. How can we get 3 scientists who are all working on Niemann-Pick type C, who have complementary skills, who all want to collaborate, who have already agreed to collaborate in real time? How do we actually get them to do it? And the answer was money. And it was a very practical issue for them, and it was a very practical issue for us. How do you go out and raise money? Now this was the really important thing that it took me a little while to realize. You don't have to raise all the money that any particular investigator needs. Incentives can be, you want to incentivize somebody and then leverage that incentive so that you get a lot more out of it than what you're actually paying for, right? So we give in SOAR, which is this collaborative of scientists working together around Niemann-Pick type C, we give, I don't want to say small grants, but we don't give NIH-sized grants to them, but each one of them is already getting a bunch of NIH money and other money. And when we give them our grants, we say, we want you to collaborate in real time around everything you do with Niemann-Pick type C. In fact, on some level, that's all we care about, is that you will collaborate with each other. And I guess the last thing that I'll say is, it was really valuable to try and think in an anti-hierarchical fashion about this. Because when you think in a hierarchical fashion, then the scientists are usually at the bottom of the rung, because, the patients are way down at the bottom, and then the scientists, and then the scientific advisory committee, and then the funders, right? And so decisions about what to do were almost always made by people who were not in the trenches. And the people who should really make decisions about what to do are the ones who are at the bottom, the scientists and the patients. So the fact that we became patient, or in our case, parent funders. And we work with the researchers very, very closely. And we meet 2 or 3 times a year, all of us together. And we got rid of the idea that there should be some higher body that makes decisions about where the money goes, or whether it's as funders or as other scientists. Really helped accelerate and sort of freeze dry the process in a way that there weren't there's very little bureaucracy, there's very little politicking. It's really mostly about what the scientists can do, what they say they can do. We have very clear milestones. Do they achieve their milestones? If they don't achieve their milestones, they're the first ones to say, let's try something else. And all of that combines with, is, I guess, held together is probably the best word, by a sense of, we know why we're doing this. We are doing this for those children. That's why we're doing this. And we're always measuring our success against what is being accomplished for them. Now, having said that, I could speak twice as long about all the problems that come up, but I'll wait, and maybe they'll come up in the next session.